Good morning. My name is Pawel Kalinski. I have about 30 years experience working with dendritic cell therapies, meaning DC vaccines, intratumoral injections of dendritic cells, and adoptive T-cell therapies using T-cells. We are also interested in targeting endogenous dendritic cells, giving patients combinatorial adjuvants. We are already in phase 1, phase 2 clinical trials in half a dozen cancers. So today I would like to share with you our experience in these areas, and trying to convince you that dendritic cells are not only vaccines, they are also both live adjuvants, something that you can inject intratumorally, or something that you can target endogenous dendritic cells, and you can use dendritic cells to develop better T-cell therapies. So in 2025, dendritic cells are not yet being widely used, but immunotherapy as such is. Immunotherapy has been a game changer for us roughly 15 years ago, and at this moment it's proven principle. We do know that immunotherapy, especially checkpoint blockers, work in multiple groups of patients and can be curative. Also, we do know that effectiveness of chemotherapy, radiotherapy, and other cancer therapies involve immune effects. Despite this massive progress, there is a massive unmet need. It can be grouped into two different areas: primary resistance, which is something where in many patients still the checkpoint blockers work well in those highly infiltrated hot tumors which have got high numbers of cytotoxic T cells into them, but they are not effective in cold tumors. And unfortunately, most of the common solid tumors such as colon, prostate, breast, ovarian, pancreatic cancer, brain, melanomas, and lung cancers show only this small prevalence of those hot tumors which can respond to immune checkpoint blockers. The second group of problems is secondary resistance. So effects typically were off in up to 85% of immune checkpoint blockers, patients who receive these therapies. Also, in most of the studies showing application of adoptive T-cell therapies with CAR T-cells in solid tumors, people see recurrence typically within weeks, more often than within months. So these challenges that we need to deal is first, immediate entry of effector cells, into solid tumors, which I believe is responsible for primary resistance to therapy. And second is antigenic heterogeneity and adaptation of solid tumors, which can contribute to both primary and secondary resistance. The reason we are interested in dendritic cells, DCs play a role in all five phases of immunity, which is important for patients' clinical responses. They play a critical role in induction and expansion of tumor-specific cells, both endogenous DCs, they pick up tumors spontaneously and induce systemic immunity. We can use as DC vaccines, and we can use also inject dendritic cells intratumorally to convert tumors into mini lymph nodes, or we can use dendritic cells in vitro sensitization, load them with antigen, co-culture them with patients' autologous T cells, and inject not dendritic cells and inject T cells. They are also very important for entry of cytotoxic T cells into tumor microenvironment. Work from Tom Gajewski's, Stefanie Sprangler, and many others have shown that endogenous CDC1s are main sources of chemokines, and they are essential for attraction of both spontaneously occurring and vaccination-induced T cells. And finally, once the T cells can move from circulation into tumors, something needs to keep them alive. And endogenous dendritic cells, again, endogenous CDC1s, other types of DCs, or intratumorally injected therapeutic DCs can play that role, working together with adoptive T-cell therapies, with immune checkpoint blockers, and with vaccines. Immunotherapy, as I mentioned, is something that we use consciously when we want to give patients immunotherapy, but it's very clear that also radiotherapy, chemotherapy, and targeted therapy all depends on immune phenomena. In the absence of active immune system, those other treatments of cancer don't work, and there is ample evidence that combinations of these treatments with immunotherapies can be beneficial. The appeal of dendritic cells is several-fold. First, we do not need to identify unique mutated antigens. Rather than sequencing patients' tumors and synthesizing individual epitopes, we can give patients, load patients dendritic cells with autologous tumor material and allow crosstalk between dendritic cells and T cells to pick up which are the immunogenic epitopes which can be presented in these patients, which are the epitopes which do have a proper repertoire of T cells. And this 6 weeks of bioinformatic and biotechnology work can be replaced, at least in theory, by simple interaction between two cell types. Second, we can use them in multiple modes of application. As I already mentioned, we can load dendritic cells with peptides, proteins, and whole cancer cells, again, inducing polyclonal responses, inducing multi-epitope-specific CTL and Th1 responses, both against mutated tumor-specific antigens and also unmutated but overexpressed tumor-associated antigens or TAAs. Also, the use of the dendritic cells does not require elimination of patient's endogenous immune system, so there is no cytotoxic fludarabine involved, making it safe and making it easy to expand. Of course, none of it is valid if unless dendritic cells can work, and I think that we are not there yet, but progress has been dramatic. On the left-hand side, there is data from 2 clinical trials. It's other data from Bidendrion in early 2000s, showing that yes, in this large cohort of patients, there was roughly 4.1 months advantage of survival of patients with castrate resistant prostate cancer. Moving 13 years forward, that's the data from ovarian cancer patients, where standard of care results only in 22.1 months of overall survival, but patients who received best standard care and dendritic cell vaccine loaded with 2 allogeneic cell lines. In this case, we got over 60% prolongation of survival. I think we're moving in the right direction. Not all of dendritic cells are made equal. Here is data from our paper from many years ago, looking at IL-12 induction in different subsets of dendritic cells. They are all dendritic cells generated from monocytes. Here are immature dendritic cells. When you look at simulation of the cells in neutral conditions, just with CD40 ligand, which mimics interaction with CD40 ligand expressing T helper cells, you do see that there is wide bar, very little of IL-12 being produced. It can be enhanced by 2 orders of magnitude in the presence of interferon gamma. Unfortunately, there's very little interferon gamma in patients with advanced cancer, but there is lots of prostaglandin E2 and other suppressors of IL-12, which can shut down this production of IL-12. For these reasons, early clinical trials used fully mature dendritic cells matured in the presence of prostaglandin E2, PGE2, and the rationale for using them was we knew that dendritic cells, when they are matured, can no longer be suppressed by cancer-related factors. They express very high levels of costimulatory molecules. Problem was that was identified in our 2000 paper that prostaglandin E2 present during the maturation completely shuts down interleukin-12, and interleukin-12 is the factor which is critical to induction of functional CTLs, Th1 cells, and NK cells. Doesn't matter how you stimulate those cells, you do not have any interleukin-12. And in accordance with this lack of IL-12, this early clinical trials all uniformly failed. So the trick we started using already in early 2000s was to give this interferon gamma during the maturation. And then we discovered these 2 phenomena which made it to textbooks since then. When you mature dendritic cells in the presence of interferon gamma, they remember the signal. So they do have memory very much the same like T cells. Then you can restimulate them with CD40 ligand, either in the absence of any additional factors, with additional interferon gamma or with additional PGE2. But please note, it really doesn't matter. What matters is what dendritic cells have seen during those 12 to 48 hours of maturation. And then they can translate this knowledge to interaction with CD40 ligand expressing cells to produce those high levels of interleukin-12. So over the next years, we have developed upgrades of those dendritic cells. We call them type 1 polarized dendritic cells. We mature them in TNF alpha, IL-1 beta, poly-IC, interferon alpha, and interferon gamma to induce very high levels of interleukin-12, IL-18, and other costimulatory molecules, promote cross-presentation of antigen from whole cancer cells to induce multi-epitope-specific CTLs, to modify their chemokine profile so they can produce high levels of CCL5 orantes, attracting endogenous dendritic cells, selectively attract CTLs, Th1, and NK cells by virtue of producing CXCL5, CCL5, CXCL10, 9, and 11, and attraction of naive and memory cells and other mature cells, okay, by CCL19. Those cells, however, as you can see here, okay, produce very high levels of good chemokines compared to prostaglandin mature cells, but produce much lower levels of the bad chemokines. Okay, it is Treg attractant, CCL22 or MDC. One round of in vitro sensitization when you are co-culturing dendritic cells loaded with antigens, in this case MAT1, GP100, and tyrosinase, okay, with autologous CD8 cells, okay, can allow you, okay, between, okay, 6-fold better, okay, numbers of functional CTLs to, okay, 70-fold better, okay, numbers of functional CTLs compared to standard DCs, which are grown in parallel cultures. And here is, okay, a list of papers, okay, where we published, okay, this data. So first, okay, 15 years okay, working with DCs, we're working okay, with vaccines. Vaccines means okay, that we are loading dendritic cells ex vivo with either whole cancer cells or with synthetic antigens, either peptide or protein. I will very briefly okay, show you, okay, this is most okay, published and move on. But here is, okay, in the second part okay, of the presentation, I'll be talking, can we okay, target endogenous dendritic cells? Can we take some of those 5 factors which we induce okay, dendritic cells ex vivo? Can we target endogenous dendritic cells okay, to promote production of those good chemokines okay, in the tumor lesions without affecting okay, other tissues? And can we do it okay, with second level of selectivity, namely without inducing okay, bad chemokines? It is possible. In the third part of my talk, okay, I would like to, okay, which will be divided okay, into 2 parts, okay, will be can we use dendritic cells in vitro sensitization to okay, co-culture okay, dendritic cells loaded with antigens, in specifically with whole cancer cells, with autologous T cells, expand them okay, for okay, 2 to okay, 4 weeks. Okay, typically we do dendritic cell assisted in vitro sensitization, followed by classical CD3, CD28, or just OKT3 rapid expansion okay, protocol. And in the final part of the okay, talk, I will tell you how can we use lessons from DC biology to develop better CAR T cell okay, therapies. So first about the vaccines. This was okay, data okay, from our latest okay, DC okay, vaccination trial. It was run by Walter Storkus and okay, John Kirkwood in Pittsburgh. Okay, okay, loaded okay, dendritic cells with 6 okay, peptides corresponding to tumor blood vessel antigens, 3 antigens targeting pericytes, 3 antigens okay, targeting endothelial cells. And that was done in most difficult setting of the disease in patients who have went through all other options, okay, including they were pre-treated with checkpoints, especially with PD-1, but most of them got both PD-1 and CTLA-4, and they still progressed. And as you can see, even if you shift from PD-1 okay, blockade okay, to DC vaccine, okay, especially this DC vaccine targeting tumor blood vessel antigens, you do see this okay, very significant okay, number of objective clinical responses. Okay, and what was particularly okay, interesting to us is that patients with primary PD-1 resistance showed all of the responses. 4 out of 7 patients with primary PD-1 resistance, we got this nearly 60% of objective response rate. In patients with secondary PD-1 resistance, there were only 2 stabilization of the diseases and no objective responses. Best predictors who responds, who not, was first primary PD-1 resistance, second response to tumor blood vessels induced by the vaccination in the blood. But there was something much more interesting. We did see okay, that in all of these patients who responded, much earlier on, there was formation of tertiary lymphoid structures in the tumors. So, okay, vaccine was converting this okay, lesions of the tumors into mini lymph nodes, and that was associated in with massive epitope spreading. So we are immunizing against blood vessels, but we do see responses against okay, lineage-specific antigens. Currently, okay, Igor Puzanov at Roswell Park has recently started phase 2 study, which is focusing on primary PD-1 resistance. In this case, we do not okay, stop PD-1, we continue PD-1, but just add vaccine. Very okay, curious. Hopefully in 2 years, we'll be able to tell you okay, how okay, things pan out. I would like to switch gears. Okay, so we do know that those alpha type 1 polarized disease produces very high levels of chemokines. Can we okay, use the same factors, but rather than treating okay, disease ex vivo, infuse them okay, in vivo, first without okay, causing massive toxicities, okay, second okay, achieving selectivity of action. We don't want to create cytokine storm. Can we do it in a way okay, which would selectively activate tumor microenvironment, okay, but not healthy tissues? Here is the data. So when we're looking at this specific problem, okay, why okay, this chemokine milieu is so important? Very early on in our paper from 2012, we did realize that CCL5 rantes and CXCL10, IP10, or CXCL9 MYC, which is not shown here, are the best predictors of which the tumor samples are being infiltrated with CTLs and which are not. Please note that this is a logarithmic scale spanning 6 orders of magnitude. So there are really very hot tumors and very cold tumors in the same group of patients, all metastatic colorectal cancer. The same data, very similar observations were made 2 years later by Jimenez Sanchez. Is that even in one specific patient, one individual patient, you have back-to-back, that's at the bottom of the screen on the right-hand side, those red hot tumors, and next to them, there are other lesions in the same patient who are very cold. When you look at the top right, you do see that even at the single site, those tumors can become hot, then become cold. In response to different treatments, they do adapt. What can we do about it? So Ravi Muthuswamy in my lab made that observation that when we are looking at dendritic cells, they attract different numbers and different types of immune cells. Alpha DC1s, here is basically shot from the top of the migration chamber. You can see that at the bottom of the chamber, alpha DC1 supernatants attracted those very high numbers of cells. Total. Standard DCs don't attract so many cells, but when you look at the bottom right, you do see that alpha DC1s do not attract any of those FOXP3 positive T cells, which are present in the top chamber of the top portion of the migration chamber. However, standard DCs, which are very poor in attracting cells in general, but are selectively attracting those FOXP3 positive Tregs, explaining why these cells have failed in early clinical trials. When we try to mimic this, of course, we would prefer not to give patients 5 different factors, including such toxic one as TNF-alpha or interferon gamma. Can we reduce it to fewer number of factors which are non-toxic? And data from another paper of Ravi Muthuswamy and from Mary Nicole Theodoraki several years later showed the same. You can combine poly-IC, toll-like receptor ligand in this case, with interferon alpha, to achieve this very strong synergy. As you can see, each of these factors by itself is very inefficient in inducing back of good chemokines. However, together they are extremely effective in synergizing, inducing very high levels, in this case in cultured human myeloid cells. What you see at the bottom of this panel, even more important, this combination induces very high levels of the good chemokines, but blocks the expression of the bad chemokine CCL22, telling that it acts potentially selectively, promoting attraction of the good cells, but reducing attraction of the bad cells. I will show you in a moment that this is indeed the case. When we try to fine-tune this cocktail further, we realize that addition of indomethacin slightly upregulates the production secretion of the good chemokines CXCL10 and CCL5, but completely shuts down CCL22. That's what happened in isolated cells in culture. Would it also be the case for a whole tumor samples, which when we can target endogenous myeloid cells, including CDC1s? To do it, we adopted this system of whole thickness tumor explant cultures. When we culture those 1 millimeter cubes at the interface of fluid and air for several hours to several days, in our case, we typically do it for 24 hours. Advantage of this system is that we have got all components of tumor microenvironment. It's all human system without any mouse components. No tissue dissociation, digestion of isolation cells. So it's simple, no cell activation and not cell losses. Results are obtained within 2 days, and 100% success rate. It's much cheaper and much more reliable than PDXs, or much cheaper and much more reliable than organoid cultures, which sometimes take 6 weeks or more just to be set up. So here is the data. As you can appreciate it, when we used this PET wells, in each case it was 7 different tumor pieces from patient 1 to patient 5. Back to back, we cultured them for 24 hours in the absence of any treatment or combination of poly-IC and interferon alpha. We do see that it doesn't matter where we started, whether those tumors produced any IL-12, any CCL5 or any CXCL10, okay, to start with, okay, we did see, okay, this massive, okay, induction to very similar levels, okay, of both of these good chemokines. And in every single case, we did see suppression, massive suppression, sometimes even elimination of the bad chemokine, okay, CCL, okay, 22. When we are looking at, what is the optimal TLR ligand, especially TLR3 ligand, okay, that we are moving forward to clinical trials, we compared back-to-back, okay, poly-IC and rintatolimod. Rintatolimod is destabilized, okay, double-stranded RNA, okay, which we didn't realize, okay, will be important. It turned out to be very important. When we are combining, okay, looking at how these two molecules, okay, can be activating two sensing, okay, mechanisms of double-stranded RNA. One, okay, dominant is TLR3 ligand, okay, the other one is MDA5 helicases, okay. We noticed, okay, that, okay, both of these factors, okay, both, okay, rintatolimod and, okay, poly-IC are activating very well interferon beta, okay, pathway. Is this pathway mediated by TLR3, TRAF3, and IRF3, okay. However, when we looked at what happens when we were interested in the degradation of IKB, it's this pathway mediated by MDA5 RIG-I helicases, we noticed that exclusively poly-IC, but not rintatolimod, is able to activate NF-κB. Here is, okay, just, okay, additional picture showing that poly-IC induces this nuclear translocation of NF-κB compared to non-stimulated cells, but surprisingly, okay, rintatolimod did not do it at all. Here is the same data, which is, okay, quantitated by image stream. We see that this colocalization of nucleus and NF-κB only occurs in the presence of poly-IC, but not in the presence of rintatolimod. And this turned out to be very consequential. Okay. So, sorry, I think I missed, okay, one slide. I'm sorry. I will, okay, missed, okay, that, okay, one slide. Okay, in brief, okay, what it means, okay, that rintatolimod, okay, is inducing higher levels of good chemokines, of MYC, IP10, and ITAC, but is not able to induce TNF-alpha, is not able to induce, okay, CCL22 or prostaglandins, those pro-inflammatory and immunosuppressive, okay, factors. So my colleague, okay, who is leading, okay, our efforts in, okay, breast cancer immunotherapy, Dr. Shipra Gandhi, took this combination to the clinical, okay, trial. She treated, okay, together with, okay, our other colleague Mateusz Oprychal, okay, currently in Indiana, okay, patients, okay, with 2 weeks of chemokine-modulatory regimen. Patients undergoing, okay, were undergoing biopsy on week 1. Then, okay, in, okay, the next week, okay, they were getting Monday, Wednesday, Tuesday, and Wednesday 3 doses of interferon alpha, followed by rintatolimod. Okay, same, okay, okay, thing. Okay, next week, Monday, Tuesday, Wednesday, biopsy 2, and patients did have option of getting pembro as a follow-up treatment. We do know that in order to achieve, okay, maximum, okay, benefit, okay, all of these factors need to be given at the same time, but for safety reasons and to be able to tell, is CKM, okay, able to promote, okay, CTL infiltration, we didn't add, okay, rintatolimod, okay, to this, okay, okay, period of treatment, which is important. So question was, okay, here, is there immune, okay, effect? Primary predominant endpoint was changes in the levels of CD8 alpha in those risk biopsies comparing week 3 to week 1. Here is what happens. Treatment was very well tolerated and primary endpoint was met. We did see average of 10-fold, okay, increase in, okay, markers of cytotoxic T cells, okay, in the tumors, CD8 alpha to the lesser extent, okay, granzyme B. There was no increases in FOXP3, so the ratios of of CD8 alpha to FOXP3 and granzyme B to FOXP3 were very highly elevated. That was associated, okay, with increases in intratumoral expression of both CXCL10, IP10, and CCL5, okay, around this, okay. Those, okay, changes were associated, I'm sorry, I'm going too fast, okay, were associated, okay, with decreases in CD8 alpha in the blood, okay, circulation, telling us that most likely what happened, we're seeing transition of those, okay, T cells from circulation to tumor. Here's what happened clinically. Overall, okay, as I mentioned, it was not our goal, okay, to combine with, okay, pembro. So, okay, clinical activity was not, okay, our goal, but we still did see something which was pretty encouraging. After 4 years of follow-up, okay, half of the patients with metastatic triple negative breast cancer were still alive. We did see one near complete responses. Here is the swimmer's plot showing all of those not capped patients have been alive at the time point of submission of this paper last year at least. And you see that at least one of them got this massive clinical response. Here is how it looked like. In these patients already after CKM itself, biopsy pre did show there was no longer viable tumors. It was only scar in this case. You didn't see much happening microscopically. This tumor is still here. That's after CKM 6 doses, 2 weeks, and 1 single dose of pembrolizumab. Sorry, that's before. Here is what happened after 6 doses of CKM and 1 dose of pembrolizumab. You could see that those tumors start to shrink, those lymph nodules. However, overall, there is pretty strong expression of pretty strong mass of tumor. However, after 3rd dose of pembrolizumab, this patient completely shrunk her tumor to undetectable. And there was minuscule lesion which needed to be resected 3 months later. Since then, patient did not require any other treatment. Becoming tumor free. This is a trial which we are running right now. We are comparing the sequential treatment of CKM followed by pembrolizumab. That was done with new interferon alpha. Merck no longer produces interferon alpha for US markets, so we started importing interferon alpha from Argentina. The generic name is Bioferon. It can be obtained, and so we are using that under as experimental drug under IND. I will be switching gears, and trying to convince you that dendritic cells can also be used as a way to deal with this tumor heterogeneity. Big problem is that typical CAR T-cell therapy. There were those 2 papers in May last year with GBM showing both the same story. First, basically infusion of CAR T-cells targeting epithelial growth factor or IL-13 receptor is able to melt most of the tumors. The problem is that very quickly later, these tumors are regenerating. They become resistant to further treatment, and unfortunately, these patients are progressing and die quite fast. Here is the data. Please note that stable disease. In some patients it was pretty significant. 12 months to 8 months, but in most of the cases, this period of stable disease was counted only in weeks. Or was not present at all. This adaptation is a real deal. So obvious reason why is heterogeneity of tumors already at the beginning of treatment, and that's key reason why Adjuvants and several of the other companies either got approvals or are in the process of obtaining approvals of polyclonal expanded TILs to be able to deal with this tumor heterogeneity. So our question was, since we know that for treatment of the solid tumors we need this polyclonal responses, can we use dendritic cells, load them with antigen, and expand them to induce those polyclonal T cells without the need to obtain sterile tumor material, or trying to find the tumor in the first place in cancers such as advanced prostate cancer, when you can have PSA in the level of several hundreds, but you cannot see macroscopic disease. Here is the data. So we are not yet in clinical trials here, just to show you some of the experiments we did with Carolina Paluca and Jacques Pancherot a long time ago, when we are using DCs loaded with tumor cells, and after 2 weeks, we are looking at induction of immune responses to different specific epitopes of prostate cancer. So we load with whole cancer cells. We don't know what we can induce in the same patients, but then in each individual patients, each of the pairs of these lines of the symbols is one patient in vitro sensitization, 2 week long. Please see, good news was that in every single case, in every single patient, we do see the immunization to against at least one of these responses. This is moving pretty well. Also, what I very briefly would like to show you this slide. Here are the advantages. And the advantages. We do expect that this alpha DC1 assisted in vitro sensitization combines both of these 2 worlds of adoptive T cell therapy using TILs and CAR T cells. We do see that we are starting with primary T cells. We don't need TILs, which are already immunosuppressed. We are targeting multiple tumor-specific antigens. We don't need sterile TILs. We don't need genetic engineering, and we are having very high expansion of young CTLs, which are not immunosuppressed. We don't need high dose file to expand them. Also, susceptibility to antigen loss expectedly should be higher. It's a definitely forward-looking statement. We don't have data from clinical trials, but we do know we are inducing hundreds of responses. When we look at TCR-D beta sequencing, and because it's basically a procedure, we are loading each patient's dendritic cells, which each patient's tumor is a procedure. So hopefully it will be approved for one indication. Obtaining approvals for other indications will be very easy, similar to TIL therapies. In the last part of the talk, I'm going to show you a few slides how we can use this unique biology of T cells activated by type 1 polarized dendritic cells to develop better CAR T cells. Bowen Dong in my lab made this interesting observation when he used one of the specific clone 40 obtained from Pedro Romero. It's the same clone, recognizing the same low level of MART1 presented on artificial antigen presenting cells. Only difference was one of this, half of this cells of T cells received pre-stimulation with standard DCs, half received pre-stimulation with alpha DC1s. Please note how much better are responding T cells pre-activated by alpha DC1s to those low doses of MART1. So what's going on? When Bowen started looking at the surface of those cells, he realized that MART1 specific cells activated by MART1 cells, not only there's massive expansion of the tetramer positive cells, but these cells are having significantly higher level of NKG2D and DNAM1 expressed on their surface. This is transient phenomenon, but at least during the several days of their activation, you do see the stable phenotype, high levels of T cells, which express not only T cell receptor, in this case T cell receptor being able to recognize MART1, but also high levels of NKG2D and DNAM1, classical NK cell receptors. When you are comparing the impact of this NK cell receptors on T cells, in recognition of cancer cells which express very high levels of MART1, there is really none. However, when you are moving to the cancer cells which express lower levels of the MHC MART1 complex, you do see that especially in those cells like MEL625, which express very low levels of MART1, suddenly the blocking of NKG2D DNAM1 abrogates most of this recognition. So this tells us that by pre-activating T cells with alpha DC1s, we are inducing this double pathway, Boolean pathways of recognition, when the cells can be using T cell receptors and then NK cells to recognize only cancer cells, but not healthy cells, because healthy cells do not express NK cell receptor ligands. So what can we do with it therapeutically? First, obviously, is adoptive T cell transfer as TIL alternatives. But second thing that we are toying with is creating a construct. In this case, there is a construct which expresses not only transgenic TCR, but also expresses those two constructs, DNAM1 and NKG2D. And when you look at what these cells are doing, when you are using as a T cell source, transgenic NY-ESO1, you do see that those TCR transgenic cells are eliminating cancer cells. Most of these cancer cells are dead after 4 hours of cultures, but 20% of them are alive. When you are using the same TCR, but also co-transduced T cells with DNAM1 and NKG2D, you do see that there is significantly reduced the fraction of the surviving T cell of the surviving melanoma cells. Sorry, not melanoma cells. They are in this case colon cancer cells expressing NY-ESO1 compared to the paternal clone. So we do believe we may have we are still need to perform in vivo studies this unique system, new type of CAR T cells or new type of TCR transgenic T cells, when we can have this Boolean and logic gate when only the T cells which express antigen receptor ligand, so peptide MHC complex, and high levels of ligands for NKG2D and DNAM1 can be recognized, but healthy cells, which oftentimes express very similar levels of such factors as NY-ZO1, MART1, tyrosinase, will not be seen simply because they do not provide this second recognition pathway. I would like to stop here and thank all of my colleagues from my lab at University of Pittsburgh Cancer Institute, Hillman Cancer Center, Roswell, our international collaborators from other centers here in the US, but mostly patients and the families for participating in these studies. I also would like to thank National Cancer Institute and Department of Defense for in excess of 50 million of peer-reviewed federal funding to bring us to this place. Thank you very much, and we are looking for collaborators in all of these areas, especially for PIs interested in moving our stuff to clinical trials. Thanks a lot.